Welcome everybody to the annual Puri lecture. This is a highlight, I know, for many of us in terms of our academic calendars for a couple of reasons. One is it allows us to usually hear some really compelling, interesting talk, and we'll get to that in a few moments, from a highly established and internationally acclaimed speaker today. And the second is it gets us a chance to catch up with the Puri family, who many of us in this room have cared for Mr. Puri for a long time. It was a team effort. Everybody worked together to look after him, and the family is like family to us. And I think that that's an important principle of why this is such a well-received lectureship. As you can see, the line is still halfway down the hall outside for people coming in, and this room is pretty full. I think lectureships like this are really important because they are a way to memorialize an individual's contributions, and you'll hear more from Dr. Puri about Mr. Puri and about some of the things that he did and why we all found him such a compelling individual. But also, I think it's an opportunity to bring people together, and these kinds of opportunities would not exist were it not for the generous donation from the family to really make this happen. We are incredibly grateful for everything that you guys have done on behalf of trying to really address the issues at the heart of mantle cell lymphoma, to try and get better at treating mantle cell lymphoma, and I think we are. Some of what we're going to hear today is about not just yesterday and today's technology, but about tomorrow's technology and approaches to mantle cell lymphoma. So with that, I will stop and have Dr. Puri come up and do her introduction. And again, welcome, and thank you again so much to the family.
Good afternoon, and welcome to 5th Surinder K. Puri Memorial Lecture. I am Radula Puri, I'm Surinder's wife. To begin with, I want to thank Barbara and Jennifer for helping us organize this event. They always do such a fantastic job. You all have heard in the past, my husband was diagnosed with stage 4 mantle cell lymphoma out of blue. And as devastated as we were with this devastating diagnosis and overwhelmed and trying to decide where to get best state-of-the-art treatment that would help him. And we were fortunate enough to end up at Winship and under the leadership of Dr. Loniel and his wonderful team. I believe Surinder got state-of-the-art best treatment, which allowed him to eventually be in remission. He had 10 years of wonderful life. He worked, he traveled, enjoyed his family, did everything that was fun. And unfortunately, in 2014, he relapsed, and he was back under Dr. Loniel's care and had almost gone back into remission and was doing all the things he loved to do till one unfortunate day in 2015, he succumbed to fulminant septicemia within hours. We have been so grateful and indebted to Winship, to Dr. Loniel and his wonderful team. We do believe that Surinder got the best and state-of-the-art available treatment that allowed him to have 12 wonderful years after his diagnosis. We have felt so indebted and really indebted to Winship and Dr. Loniel that we as a family wondered what little humble gesture we could make that would allow to continue to improve the care of patients of mantle cell and B-cell lymphomas. And that is what originated this humble gesture from our side, so that we could bring international researchers like Dr. Rimsza and collaborate and do multi-institutional collaboration, which I have understood have been going very well. So thank you. I do want to say a little bit about my husband today, not as a patient. We've talked about all the things he loved to do, but I want to talk a little bit about him as a person today. I was married to him for 45 years, and it was very clear to me right in the beginning that I was very fortunate that it was important to Surinder that not only his profession took off and he did well, but it was important that my profession also would be equally important, and he did everything that I ever could imagine to help me have my professional life. Surinder was working, it was the 70s, he was working for an international, one of the biggest international architectural and engineering firms in their corporate office here in Atlanta. It was the 70s, and a lot of young architects and engineers were entering the field of corporate world, and which was primarily dominated by male professionals. And I have heard from some of his younger colleagues and friends, sometimes it was intimidating, sometimes it was disheartening, and somehow they would find their way to Surinder's office for his listening ear and his encouragement and support that he offered women. It was very important to him that women professionals enjoy the freedom that all men have done. And he was unique, I think, for my experience in that sense. So I really wanted to share that, especially today, because not only we have an international researcher here, we have our first woman international speaker in this series. And I am so excited to welcome Dr. Rimsza for that. Surinder's motto, I think, in life, from my point of view, always was, you too. So I just wanted to share that and welcome you, Dr.
All right, thank you very much. And we'll go ahead and move forward now, and I'm going to briefly introduce Dr. Rimsza. We're very fortunate to have her here from Mayo, Scottsdale. Dr. Rimsza is an international expert in lymphoma biology, pathology. Her research programs include a number of different areas, including immune evasion in lymphoma, oncogene dysregulation in lymphoma, and she's done a fair amount of work in HIV-related lymphoma as well. What I'm really actually enjoying about this is that it's rare that I get a chance with really good friends that I have to name drop somebody from their own institution. And the myeloma group at Mayo is pretty active. We know them very well. And I'll be able to just say, yeah, we had Dr. Rimsza was here the other day, and then just leave it at that. And then I am certain they're going to call you and say, are they trying to recruit you? So play this up for all you can get. We're very glad to have you here today and look forward to your lecture. Thank you.
I'm really super excited to be here, and I can't tell you, they invited me 2 years ago. My daughter was getting married, so I could not come on that day. And last year you invited me again. I was going through some medical treatment myself. Notice the long black hair in the picture and the short gray hair now. But anyway, so this year, third time's a charm, and I got to meet the family last night, and you guys were just so personable and so much fun, and it really just brought so much meaning to, you know, what we're doing today. And again, it's been so well planned. I have to just thank the team that put this together. Everything is run on time. Everyone's been friendly and enthusiastic, and I guess this warm Emory welcome has been really overwhelming. So I just, again, thank you so much. So I am a pathologist. The last 4 people you had coming through were a hematologist, oncologist, medical oncologist treating patients. So I'm going to have a little bit of a different perspective coming from the tissue diagnostics end of the spectrum. And I wasn't sure what the audience would be, and I thought we might have a pretty broad audience. So I'm starting really at the beginning, and some people may say that's ancient history, but hopefully I'll bring you all the way up to what we're doing and what we're working on today, and so it'll make a really good story. So I called it Mantle Cell Lymphoma, a quest for biologically defined diagnostics and prognostics. And we just touched on this, Dr. Lanell, about it should be biologically defined, not based on appearances. So, lymphoma, okay, malignancies are the adaptive immune system, first used in 1858, and it was just one disease, a big lump, a lump in a lymph node was a lymphoma. We know that they're malignancies of B's and T cells now, and we classify them based on the type of B and T cell they come from. And as our knowledge of immunology has improved and our knowledge of genetics has improved, we've continuously refined our categorization. Now non-Hodgkin's lymphoma has more than 85 types. So for those of us in HemePath, we always say this is our job security, right? Because we keep making more diseases, it's more and more complicated. So I'm going to, like I said, start at the beginning. In 1966, Henry Rappaport came up with a classification for lymphoma. And we didn't know much about the cells. We could just look at them under a microscope and we can say, gee, they're round or they're not round, or they're big or they're small. And so I just have a picture here showing, round cells on the top, irregular cells here, and then large cells here. And we thought these are poorly differentiated, these are well differentiated. These, we called them histiocytes because they were big and we didn't know that lymphocytes could get that big. We also looked at the growth pattern, and we could say something is growing in nodules. This is showing nodules of cells, or there's growing in a diffuse sheets of cells. So those were the parameters that we had. So in 1966, we just had a purely descriptive diagnosis of a classification for lymphomas. And you can see the terms here: nodular, poorly differentiated, nodular, mixed, nodular, histiocytic, diffuse, blah, 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 blah. Where in 1966 would mantle cell lymphoma fall? Probably would have fallen mainly in this diffuse, poorly differentiated lymphocytic lymphoma. You might have had some nodularity to it. You might have had some large cells in there, which we would now call pleomorphic variant, and you might have had some blastic ones that would have been called, now we know those as blastoid mantle cell lymphoma. So they were mixed up in here. So as you tried to study this disease, there was really no way to study mantle cell lymphoma because you couldn't even identify it. The technical leap came forward was immunohistochemistry, and at the time, when it started, we only did it on snap frozen tissues. This was done at academic medical centers such as yourself and other places, monoclonal antibodies applied to tissue, being developed into different colors. And we could finally just see that there was a mantle zone here. This is the CD20 highlighting the B cells. We have germinal centers. Here with dark CD20, and then we have these lighter cells out here. This is a normal lymph node, but we could just see that these cells had a little bit less CD20 and some other features as time went on. So we were finally able to say, hey, we do have something called a mantle zone. In 1973, then, there were a couple of warring lymphoma classification schemes. There was a group called Lukes Collins. They were at Vanderbilt, Kiel, and Kiel, Germany. And they incorporated B and T cell information. One was widely used in the United States. The other one was used in Europe. The difficulty there was because you had two classification schemes, when you were trying to compare and integrate information from across, the Atlantic Ocean, you weren't sure if you were comparing apples to apples or apples to oranges. The other thing is that neither of them really translated into all of these patients have this kind of prognosis or outcome because they were too crude. There were a lot of different diseases in each of those big categories. So we still had a definition problem. Some people say a step forward, some people say a step back, but in 1982, go fast forward 10 years, we had the working formulation. And this was based on the clinical outcome. And this was driven by the clinicians who were trying to do clinical trials in patients with lymphoma and trying to use Lukes and Collins and realizing if we put patients on trials based on these sorts of definitions, we don't know. Some of them probably had a poor prognosis to begin with, and some probably had a good prognosis. So what we're going to do instead is look at all of the data, and we're going to take groups of patients that we think have a similar prognosis, and we'll put them together. So kind of going away from the biology and going to the clinical outcome and putting those together to do clinical trials. So we had low, intermediate, and high risk based on survival statistics, and then going back and saying, oh, what did they look like? So each group had more than one morphologic category. There was no reference to the immunological origin of the cells. It was very popular for clinical trial design and probably dominated for about 15 years. This is what it looked like in 1982, low, intermediate, and high grade. 5-year survival statistics, 50 to 70, 35 to 45, and 23 to 32. And you can see that in there were 3 to 4 different types of descriptors within each of the working formulation categories. And again, where would mantle cell fall? probably in the diffuse small cleft cell lymphoma. So here is when I started training. We had working formulation, Rappaport, Lukes, and Collins, and you had to be able to translate, and this was one of our jobs, was translating, oh, you have nodular histiocytic, you probably have follicular large cell lymphoma, and vice versa. And again, mantle cell probably fell in working formulation E most of the time, but again, there could be a lot of other diseases that we now call out as separate disease entities that were in there. Probably, if it's in the mantle zone pattern, some could have fallen here. The pleomorphic variant could have fallen here, and again, lymphoblastic could have fallen here. So then as we started to move towards a definition of mantle cell lymphoma, we had to kind of back it out of the working formulation E. Working formulation E had a bunch of different diseases probably embedded in there, and there was a series of papers. I just listed 3 at the bottom, and I'm happy to list these. These came out of the Southwest Oncology Group, the SWOG committee, which I was later associated with, but there were many, many others. And they started realizing, as you look at working formulation E, oh, it's important if it's B or T cell. We laugh at that today, but at the time that was a rejected concept that B or T cell made any difference. Blastoid morphology was bad. Lacking BCL2 rearrangements was bad. Well, we now know that those probably, if it was there, you're identifying a follicular lymphoma. So if you didn't have that, well, then you maybe had a mantle cell lymphoma. And then they also started looking at the patterns and saying, oh, mantle zone pattern might be a bad outcome. So we started pulling out of working formulation E what maybe a definition for mantle cell lymphoma was. So it was probably not into the early 90s that mantle cell really came out with a good definition. And as I said, if you can't define it, you can't treat it, you can't test it, you can't learn biology. But we realized there was a characteristic cytology, there's a characteristic growth pattern. We found out that it often had expression of a molecule called CD5 on the surface, okay? And that was a technical advance. For those of you who remember back in the day, we didn't have a CD5 antibody that worked in paraffin, and we used CD43 as the poor man's substitute. So anyway, CD5 was a technical advance. We also found out that there was a characteristic translocation, the 1114 translocation, and that was discovered through cytogenetics, seeing that the BCL1 gene, B-cell lymphoma 1 gene, which was originally called PDRM1 because it was identified in parotid gland neoplasms initially, went next to the immunoglobulin heavy chain gene. And that means that it was overexpressed, and you got a lot of cyclin D1 protein. Cyclin D1 is part of cycling, so that's helping the cells to divide and proliferate. The patients often had extensive disease, a lot of involvement of the GI tract, and the survival of about 2 to 5 years. So not a good survival overall. The next big leap forward then was a few years later came what we called the real classification. And this was you had to get the European and you had to get the North American pathologists together in a room to duke it out and say, what is the real classification? We can't have working formulation, Keel, Rappaport, and then new entities coming out. So this was the revised European and American classification, a proposal from the International Lymphoma Study Group. And again, my mentor, Tom Grogan, was part of that group, and I've since become a mentor, so I'm very proud of this little lineage here. But it was really the origin of the modern World Health Organization classification scheme, which we all now adhere to. We all have the WHO book. We look up all the things, and it's sort of the codified Bible of the classification. This group said we've got to go back to return to cell of origin, the histogenic classification. We can't just look at survival and then back assume that we have certain diseases. We have to go back and label these diseases based on the type of B cell that they come from. And they included morphology, they included phenotype, because phenotype was now widely used, genotype, either cytogenetics or FISH, and then also clinical presentations to go together to define disease entities. So that was really the beginning of modern lymphoma classification, 1994. And there's 2 main groups, the mature B-cell neoplasms, the mature neoplasms, B and T, and then the precursor B and T. And mantle cell lymphoma fell into mature B-cell neoplasms, and there were only about 12 diseases at that point. And I'm telling you now, there's more than 65. So it was based on this, and this is a figure from the WHO showing, B cells, we know they start in the bone marrow, go to the blood, to a primary follicle, to the mantle zone that we could see, the germinal center, marginal zone, back to the bone marrow. And we have an antigen exposure to B cells. Remember, these are immune cells. Initially, they're kind of unfocused, but then once they encounter an antigen, they become more and more specialized, and the mantle cells go into the follicular area and become blasts and move on and eventually become a plasma cell. And so we decided, the REAL said, we're going to go back, we're going to label all of our diseases as coming from one of these levels of differentiation of B cells. And so obviously mantle zone B cells gave rise to mantle cell lymphoma, and that was 1994. So then the tables got worse, and this is a little bit blurry, I know, but mantle cell lymphoma is right there, okay? So actually, I started my residency a year before the REAL came out. So I only had to do working formulation for like a year. Then I was very happy to switch to REAL classification. And you can see in working formulation, they have several things, that would all be mantle cell lymphoma, and same thing with Kiel classification. So I'm happy to say mantle cell found its way, and it has remained its own disease ever since. Some of these other ones have kind of come and gone, but mantle cell has remained. So now we have the WHO 2001, 2008, and now we have 2016, but these kept all the principles of the REAL classification. We had 37 diseases in 2001, 67 in 2008, and we're at 85 plus for non-Hodgkin's lymphoma now. And again, mantle cell continued to have its own diagnostic category. So once we have a definition of a disease, and this was really 2001, before everyone started using the WHO. Before that, people pushed back against the REAL and said it's not prognostic. People had to go around the world, collect 1,000 cases, show that the REAL classification had use. Dennis Weisenberger did a lot of that, and others. And we could start now really looking at biology of lymphoma. And you can see mantle cell lymphoma over here, diffuse large B, follicular lymphoma, composite, which is usually a little bit of both of these. But mantle cell lymphoma, about 6% of all non-Hodgkin's lymphoma, and I just estimate it's about 3,000 to 4,000 cases a year in the United States. So this was the first kind of look at what we actually are dealing with. The definition continued to improve. We knew there was a predominance in male patients with a predominance around that 40 to 60-year-old age group. I have the median survival still at 3 to 5 years, but it's extremely variable. It can be shorter than that, and it can be a lot longer than that. And the clinicians in the room will tell you there are some patients that you can actually watch even for a little while before you start treating. So it's extremely variable. We knew by then that it for sure originated from the mantle zone B cells, had co-expression of CD5. About 85% had that cytogenetic change, that translocation, upregulating cyclin D1. We found that there were other cases that didn't have that, and the other 15% had other alterations of cyclin D1 or expression of other cyclin-related proteins, cyclin D2, cyclin D3. So we were getting, once you have a tight definition, then you can start saying, okay, but there are some exceptions. And there are some exceptions of cases that don't have cyclin D1, but have other proteins. And so this is what it looks like. Generally, it's a diffuse pattern. You can see these cells that are kind of round, slightly irregular. You can understand why early people looking under a microscope might put them in one category or another. We developed the cyclin D1 immunostain, which worked well in paraffin. That was a huge help. And the other thing I wanted to point out, and it's a little hard to see from here, but you can see some mitotic figures in here. When mitotic figures, when a cell's going through mitosis, of course, all the chromosomes line up in the middle and then they pull apart. And that's a measure actually of how fast the cells are proliferating. If you see a lot of mitotic figures, those cells are proliferating. And it was recognized early on that mitotic figures might have something to do and relate to how those patients did in the long run. Okay? Technical leap forward also, because I want to emphasize technology, because I'm a techno geek, was doing FISH, fluorescence in situ hybridization on paraffin-embedded tissues. Instead of having to grow cells in a tissue culture dish, poison them, look at their chromosomes, stain them, and categorize them, these are small probes to sections of DNA that can be applied in regular paraffin-embedded tissue. So, of course, when a biopsy comes out of a patient, generally it goes into formaldehyde. And as my mentor said, that's the difference between having a cucumber or a pickle, okay? So once the tissue goes into formalin fixation and paraffin, you're dealing with a pickle, okay? It doesn't have the same chemistry as a cucumber, all right? It looks like it, but it's different fundamentally. So everything I talk about, I keep saying, well, that was done on frozen tissue, but now we have to do it on paraffin. Frozen tissue, again, is not generally available unless you're at an academic center and you get a large size biopsy. You have the infrastructure to take a piece of tissue, snap freeze it in liquid nitrogen. Otherwise, for 99% of the people in the country, tissue comes out, it goes in formaldehyde, okay? So this was a big technical advance, that we could take paraffin-embedded tissue and start looking at the DNA structure. And this is just an example of a green and a red probe to two parts of the cyclin D1 gene. If that gene is disrupted, those signals fly apart. And so you can see then a green signal separate from a red signal, and that tells you you have a cyclin D1 translocation. So that was super helpful, and we use this in a lot of our studies to confirm the diagnosis so that we know when we're studying with really complicated techniques, oh yeah, we have a cyclin D1 rearranged positive tumor. So 2016 came out in 2017 actually, right? But I think we call it the 2016. And now we have a little bit more of mantle cell lymphoma description with more caveats though. So again, demonstration of the 1114 or cyclin D1 protein expression, recognizing again that rare cyclin D1 negative cases exist. We also have in there a section on the histologic appearance with the aggressive variants being blastoid or pleomorphic. Again, I'm looking at you, reliance on the morphology. There's other variants that are small cell or marginal zone-like. There's also a diffuse, a nodular, and a mantle zone pattern. So you can see, again, back to the older days where it really fell in a lot of different categories. We also recognized in that time that there are different clinical pathologic variants. So there's something now called in situ mantle cell lymphoma. And this is mantle cell lymphoma that is kind of incidentally identified. So maybe someone is having surgery for breast cancer and they get lymph nodes taken out, or they're having a cardiac procedure and they get chest or aortic lymph nodes taken out. And you look at those lymph nodes and you think, that looks a little funny. And you can do a cyclin D1 stain, and you can find a little rim of cyclin D1 positive mantle cells in the mantle zone. And if you do FISH on those, you can find the translocation. So it's a little bit of mantle cell sitting there on the inside. If you follow those people forward, a few percent will go on to actually get the disease mantle cell lymphoma, and some will just never have it. So somehow the immune system is keeping all of that in check. So we recognize in situ mantle cell lymphoma, it's an area I think people are interested in following forward and doing more work on. We also recognize another variant called the leukemic or non-nodal variant of mantle cell lymphoma, and it had been detected that sometimes mantle cell lymphoma is occurring as lumps and bumps in the tissue, but sometimes it's mainly in the peripheral blood, and you can find it circulating out there. And it's in the spleen, which is saturated with peripheral blood, and it's also in the lymph node where the peripheral blood is going through. So, in the bone marrow. So bone marrow, spleen, but usually just spleen and peripheral blood. And these patients often don't have a marker called SOX11, which is typical also in mantle cell lymphoma. So there was a thought there, are we going to call this a different disease or not? But it has remained in the mantle cell category, and these patients do very, very well. And I'm looking at my lymphoma clinicians, so when, in this case, some of these people get followed, right? They don't get treated right away. So that was a distinct group, and it has a distinct gene signature. So now that we have an accurate diagnosis, and then first we had to make it a very uniform diagnosis, then we had to start saying, well, what are all the caveats and exceptions, right? And now we're saying, okay, we want to now know if you have mantle cell lymphoma, how are you going to risk stratify? We know that patients have a really variable outcome. And so the next big step forward, I would say, was the MIPI, the Mantle Cell International Prognostic Index. And this was developed on patients treated with conventional chemotherapy. And what they found was that if you look at the patient's age, their performance status by the Eastern Oncology Cooperative Group, and blood level of LDH and white blood cell, with those 4 parameters, you could come up with an international, with a prognostic index, which is called the MIPI. There was an IPI and there's a FLIPI, and this is the MIPI, okay? And this is what the survival looked like for patients with different MIPI levels. So if you just go out here to 5 years, the patients who are in the upper level, low risk, their survival is over here around 70, just 65 or so %. If you come down to the intermediate group at 5 years, you're down a little below 50%. And if you come down to the very bottom group, it's down around 20%, all right? If you haven't seen these before, this is months, and this is survival. So when everyone's diagnosed, everyone's alive, and this is the survival curve as the patients pass away, showing the showing the overall survival of that group. So the MIPI separated these curves very well. The next thing that happened was we said, can we do better than just the MIPI by itself? So this group, led by the Germans, came up with what they called the MIPI-B, the MIPI biological, and they added in a stain called Ki-67. So instead of counting those little mitotic figures to say how proliferative is this tumor, you can look at the Ki-67 stain and say, hey, these cells are actually in cell cycle, and if there's a certain amount percentage in cell cycle, those patients will do worse. And so when you add that to the MIPI, and again, you come out here at 60, you can see that if you have a low MIPI and a high proliferation, that survival at 5 years is down around 10%. So these patients are really, facing the most difficult odds. And so it's kind of splits those curves. It made us think, well, in addition to the MIPI, there's biological things that we can count and measure that might be additive and help us better look at any one patient and say what may happen. The problem with this is, how do you count Ki-67 rates? And this is a big part of what I've been working on with mantle cell lymphoma, and it's harder than it looks. So the group from Germany came out, this is in 2009, Wolfram Klopper, and he said, how do most pathologists count how many cells are positive? Well, most people just eyeball it. They just go, yeah, it looks like it's about 30%. It looks about 80%. And if it's close to 100 or down below 20, it's pretty easy. But when you get in the middle, it's a little hard. And the significant cut points were determined to be at 30%. So this is just a graph from their paper showing Ki-67 by eyeballing versus counting. And you can see, yeah, there's a cluster down here of low cases, a cluster up here, cases that were high by eyeballing it and by counting it, but there's a lot of variation. Here's a case by eyeballing was called 85%, and by counting was down here at 20%. So, and so you can see some extreme variation in here. And so they sought to try to really reproduce the way that people did this. And so their bottom line was eyeballing does not match counting, and you should really count. Initially, they said 2 times 500 cells, 1,000, but they decreased it to 300. And I think most people don't even count 300. But another thing that's very important is the pitfalls and the selection. Where in that whole tumor are you going to stop and count how many cells are positive? And these are this is one case here, and this is another case here. And so they've done Ki-67 staining, and you can see there are big clusters here, this is low power, of cells that are positive. These are probably residual germinal centers. This is probably not even the tumor at all, okay? The tumor is in this area. So in this case, and they also point out there's some areas that have a little more, a little less. So really, they said, if you're a pathologist picking where you're gonna count, you should be counting in this area. Here's another case that is a very nodular pattern of mantle cell lymphoma. You can see a lot of positive cells down the middle, and you can see these areas here that have a lot fewer. And basically, by doing multiple immunostains, they were able to see that these are T cells, not even the tumor at all, that are surrounding the tumor, and those are proliferating. Again, you should be counting here, and you should be counting here. And so if you can see that the difference between 30, this might be 50, 60%, this might be 10. So field selection is critically important. And so when I talk to my residents, I say, follow this paper, count in this area, but I don't think this has been widely adopted. I think most people still do sort of an overall average. Okay, so we're going to stop that story there, and then we're going to technically leap forward again into gene expression profiling, starting out with frozen tissues, and then we'll go to the pickling chemistry, the post-pickle chemistry. So frozen tissues, and it can interrogate thousands of genes. I remember the first time I saw this, the Nature paper from Ash Elizaday in 2000, I was just blown away that we could interrogate, instead of one little red or green dot at a time, everything at once. So you all know DNA has a genetic code, there's 30,000 genes. You go to the mRNAs, which are the active genes, which give you the proteins. So this is the sweet spot now, is we're gonna measure all of the mRNA, all of the levels of activities of every gene in the cell. And this is basically how it works. There's a solid support. Initially it was glass slides, later it went to a little chip. You have these probes, AGCC, AGG, whatever, all these probes. You take your RNA out of your tumor, you give it a little fluorescent, you tag it with a fluorescence, and you lay it over, and then it'll line up where it needs to be if there's a matching sequence. And from this, you can then count how much fluorescence there is and say how active is any one gene, okay? And by knowing the position of these probes, you can read that like a book. And this is a little picture of an Affymetrix chip showing kind of just a cartoonish, but different little colors of fluorochromes. So this changed a lot in hematologic malignancies, and it's gone on to work in other malignancies as well. So when microarray came out, and people who were interested in mantle cell lymphoma said, okay, is there a gene expression pattern that more accurately defines mantle cell lymphoma? And are there key genes that indicate which disease, which case is going to have a poor or a good outcome? And so proliferation genes were actually a big a candidate, but it was agnostic. Are there other pathways? Are there other things we should be looking at other than just proliferation? And the answer is proliferation is a big deal in mantle cell lymphoma. In other lymphomas, it might be the microenvironment, it might be something about differentiation or activation. That early look at those little mitotic figures really was a key important observation. There's a very distinct diagnostic signature for mantle cell lymphoma. You can take cyclin D1 positive, negative, SOX11 positive, negative, nodal, blood, whatever. You can look at that, and it is a slam dunk to tell mantle cell lymphoma from diffuse large B-cell lymphoma, marginal zone, any other type of lymphoma. I'm actually not showing that here. But there was a prognostic signature predicting how well the patients would do, and all of those genes fell in somewhere in the proliferation pathway. So this is the original paper from the LLMPP, Lymphoma Leukemia Molecular Profiling Project. I have since joined them. I was not part of this original paper, but it confirmed all those previous Ki-67 and other studies, and you can see a survival difference for patients that have low proliferation versus high. These are very old cases. Survival overall is not good. These were pre-Rituxan cases. But it did confirm the proliferation signature as being very important. Now, caveat on that work, this was performed on what was called a lymphochick microarray, which was built by hand by Dr. Lou Stout at the NIH on a glass slide. And it did not actually cover the entire genome. Out of the 30,000 genes, it covered about 12,000. We also had at that point what we called an unbalanced gene expression model. We were only looking at genes that were overexpressed. We didn't look at genes that were underexpressed. And we've since learned that when you're doing mathematical modeling, it's good to look at both what's over and under and put that together. So we took these cases and we ran them again, and this is when I joined the group, on what was called the Affymetrix U33 plus 2.0 chip. For those of you in the field, you know this is an entire genome chip. It hits every gene in the genome. So 80 of those original cases, 80 of the original 92 from the first prior paper were reprocessed, and I'm going to tell you that this is now used as what we're calling the gold standard moving forward. As you're trying to develop a biomarker, you're developing technology, you have to compare it to something. So somewhere you have to have a gold standard that's sort of unassailable to compare how your new technique is doing. And this had 30 housekeeping genes, which are just genes that every cell should have on and turned on at a certain level. And then you had 69 other genes. It's important to include these housekeeping genes because then when you're comparing sets of data to each other, you can compare those and you can normalize against that. So for those of you who are mathematical know what I'm talking about. The caveat with this Affymetrix data was gold standard was it really didn't work that well in paraffin-embedded tissues, okay? So Affymetrix worked great with cell lines, cell suspension works great with multiple myeloma, leukemias, and et cetera, but most of the lymphomas are in paraffin-embedded tissues, and you get a lot of gene dropout, okay? So so how are we gonna best measure proliferation? We know it's important. We've done an agnostic look at all the genes in the genome and found out that really, yes, proliferation is really the key issue. Frozen tissue is rarely available, as I said. The Ki-67 can be done in fixed tissue, but it is really not that reproducible. And so the question is there a better way to assess the cases? So technical leap forward again comes digital gene expression profiling from fixed tissues. And we've worked extensively, and I think I'm supposed to have a conflict of interest slide here. I have an invention based on this technology, so I may have a conflict of interest. But anyway, we have worked extensively with this technology from NanoString. It works very well with paraffin-embedded tissues. We used it, and most people are more familiar with our work in diffuse large B-cell lymphoma to decide on, classify into 2 or 3 different cells of origin, and a newer one with primary mediastinal B-cell lymphoma. But we also applied it in mantle cell. We also checked Illumina, Affy, HTG molecular, and we did a bake-off of different technologies, different extraction kits. We spent time with, I think we had 4 extraction kits, 4 methods. We looked at 16 ways, and we said, this really works pretty well. So let me walk you through how it works. So here is a formalin fixed tissue block. It's in paraffin wax, okay? You cut the slide, you cut the block and you stain it so you have your tissue on a slide. We review it. We have to circle an area that is predominantly at least 60% tumor. And then we take a little, really a little razor blade, and we'll just scrape off the area that's actually the tumor, put it into a tube, and extract the RNA. We found that there's a lot of ways to extract RNA, and we have a favorite method, but several of them will do. So what's the NanoString do? And you can buy probes labeled from the company, which is a little difficult because each batch has a different fluorescence intensity. So what I do in my lab is we buy cold probes with no fluorofoam or nothing attached to them, and we label those ourselves in our lab with what's called the elements chemistry. And that has allowed us to be extremely reproducible between runs. So you have the NanoString. This is the string of reporter probes. You can see 6 different locations, and there's 8 colors. So by multiplexing that, you can come up with 800 different color sequences. So it doesn't do the entire genome. So if you've already done all your discovery work on Affy and frozen tissues, and you have a list of genes you're interested in, then you can go to NanoString and say, I'm going to apply this in a laboratory setting. And then there's a capture probe. So we have oligo probes that match up to the reporter, and oligo probes that match up to the capture tag. These are going to come down and seek the target RNA. So this is the RNA coming out of your patient's sample. And then those sit down in this method on top of the gene of interest. And so you have a little capture probe. When you run it across an electrophoretic current, that little capture probe will go down and like hook onto the bottom. And then this little thing will also lay down flat, and then you just come in and scan it and look at the barcodes, and then you can quantify the amount of RNA that was originally in there. This is what it looks like. This is the instrument that does all the pipetting and et cetera. This is the little electrophoretic cartridge. The sample goes from one well across a tiny little tube to the other well, and all of those little barcodes are captured and they lay flat. And then this is what the data looks like. And obviously you have a specialized camera that will capture this image and say, okay, here's something with a green, red, yellow. Here's another one, green, red, yellow. We have 2 copies of this. Maybe we only have 1 of these, and it's beyond me to do it, but the computer does it, and it'll say, you got hundreds of these, you got 20 of these, you got 31 of these, and you got 0 of these. And it's very quantitative, and when compared to other methods, again, it seems to be stoichiometrically related to the amount of mRNA that was there in the first place. So here comes when I started to lead this part of the project was in 2011, and we wanted to refine the diagnostic signature, again, for the cyclin D1 negative and other rare subtypes. And we wanted to see if we could improve that initial proliferation signature to more accurately characterize the patients and guide the clinical care. So going back to our seminal paper that we published in 2017, I shouldn't say seminal, our first paper in 2017, we had a training cohort of 47 cases that we ran on the NanoString. 39 of those had frozen, snap frozen tissue, fresh frozen, with the Affymetrix gene expression profiling. These were those gold standard cases that I mentioned before. So we wanted to see how these cases with the data and information we knew would perform on this instrument. From that, we selected the best genes. There were 17 genes related to proliferation, 18 related to housekeeping, for a total of 35. So you'll hear me talking about the MCL 35. That's where the 35 is coming from. And we developed an algorithm, and then we locked it, and we blinded ourselves. We're never touching it again. We're truly going to go into a blinded, independent validation cohort. So we got another set of cases, 110, pre-treatment, all lymph nodes. They were treated with R-CHOP, and some were, about half were with intent to go into autologous stem cell transplant from British Columbia Cancer Agency, and we did a lot of reproducibility. So this is a picture of the mantle cell 35, the MCL 35, refining the MCL signature. We got down from a lot of genes, 69, down to a smaller group. And these are all of the genes that we found were important. Those that are overexpressed are coming out in red, and those that are underexpressed are coming out in green. And each little column along the way is one of those 110 patients. Out of the 110 patients, 108 worked. So that was a really good showing that it really works well on pickles. So here's mantle cell lymphoma risk groups, the low, the standard risk, and the high risk. We looked at how that related to morphology, blastoid and pleomorphic. Of course, they tend to be in that high risk group. We looked at P53 staining, which has been an important topic of discussion in mantle cell lymphoma, tended to be in the high risk group. A few in the standard risk, and this deletion of a part of the CCN D1 3' untranslated region, which gives stability to the mRNA and was associated with poor outcome. But for today, we're going to focus on the Ki67 and the MIPI. So the Ki67, you could see most of the cases that were greater than 30%, the dark gray, are falling up here. And most of the light gray are falling down here. But you definitely see exceptions. You see patients here who have very low proliferation using Ki67, but are coming out as very high risk. And vice versa, you have patients down here that are in the low risk category by Ki67 that are actually high risk by gene expression. So there is some discrepancy in there. And MIPI, again, a tendency of the high MIPI people to be on this side, but various people coming out in different areas. So it seems like the MIPI is adding to the MCL 35. Okay, so using the MCL 35, again, these are R-CHOP treated patients within 3 months of diagnosis with a planned autologous stem cell transplant if they were less than 65. Here's the time, the overall survival. Here's 5 years. By MCL 35, you can say 85% chance you're going to be alive in 5 years, about 40% or about 20%. So we were really excited when we saw this, and then other people right away who were doing mantle cell lymphoma clinical trials said, we want to apply this retrospectively and look at our trials. So I'll show that in a second. What I was pretty excited about was this mantle cell MCL35 reproducibility. So if you remember back to the eyeballing, or you remember back to the counting, this is 3 different assessments, and you can almost not even see the different dots. Different cases run 3 times and falling along this line. They were run in Germany, Spain, Canada, and then I ran a subset also in Scottsdale. These are not even adjacent, these are not RNA aliquots that were sent around. These are not technical replicates. These aren't even adjacent cuts. These are non-adjacent cuts out of the same block. So this is a true biological replicate, and you can barely see any difference, and 100% every patient came out with the same result in every lab around the world. So we thought, this is great. We have since gone on, I went to the NIH, the clinical trials office. If we'd like to use this in clinical trials, we need to develop it further, and for the pathologists in the room who sign off on laboratory developed tests and go through CAP inspections, you know you need to have all of this data. We had to create standards and controls, which we did. We bought ultramer RNAs, diluted, pooled, frozen, we use as positive controls. We wrote standard operating procedures, locked those down, tested a bunch of cases. The accuracy, 92%, that was comparing back against the frozen tissue. We actually think that it should be the other way around, that the gold standard is 92, because we think this works very well. Precision, 100%. You could run one sample 40 times and it'd come out the same. Diagnostic sensitivity, as far as getting people into the same risk class. We looked at the lower limit of RNA that should go in. You can get down to just 50 nanograms, so oftentimes one slice, we even do it off of core biopsies. We did look at different fixatives. B5 fixative doesn't work. Decal also is challenging, but works about half the time. So this is from our clinical lab, mantle cell, a score here, Ki-67 here, kind of a general good, a general trend together. But again, when you look at it with a Ki-67, you can see a few patients that are low risk that end up in medium, and a few high risk patients that end up in low. So this is a not as many cases as the previous paper, but kind of shows that we have the same kind of issue going on. So after that, the Norwegian lymphoma group got involved and published a couple of papers. For those of you in the clinical world, I think you're aware of the Norwegian data showing the really hyper-intense treatment and some of the benefit to outcome. So they went back, and this is mantle cell lymphoma cases with MCL35, again, a very now familiar-looking Ki-67 and MIPI correlation. This is the survival of the Norwegian trial patients. You can see the low and the intermediate risk end up going together, and the high risk coming out separately. And so people puzzled about this at first, but what you'll see is that the low risk patient at 5 years actually are close up to 45 to 50%. So this was a younger, healthier cohort of patients. They got not just standard chemo, they got very aggressive treatment, and everybody as a whole was doing better. So what about MCL35 in the Norwegian cohort? Can we add it with the MIPI like we did with the MIPI biological before? And the answer was, yeah, we can. So here is, the low, intermediate, and high risk over time. If you add in MIPI and MCL together, if you have poor MIPI and high-risk MCL, you can see this very, very difficult group of patients to treat. And the question is, why blast them with the intense chemotherapy? So the discussion from that paper said maybe we should be considering biological risk factors with MIPI in a kind of a combined approach. It did identify that small group with the really dismal prognosis, and clinical trial recruitment for novel investigational drugs might be warranted, and they were very happy with the performance of the assay. Since then, there's been a second paper published by the Norwegians, and I just saw another paper coming from Denmark. So Ki-67 proliferation index is still being evaluated, and I think there still may be a very good role for it. This just came out in 2020, so I saw this paper 10 days ago. and I wanted to put it in just to be complete and show you where we are in the field. So this is the same German group that had done all of that about how do you count and where do you count and how do we get this done correctly. And so they had 8 reference labs. These are all academic centers in Germany and Europe. And they did a round-robin test. They sent around a tissue microarray with 20 different cases. They said stain it how you would normally stain it and count it how you would normally count it. Now, one thing I want to point out is when you have a tissue microarray, you've punched little pieces of the original block and put them in, in a single block to make it easy to send around. So there's no field selection in there, right? Every pathologist looking at it has to count the same field, so because it's already been pre-selected for them. So that should decrease variability, if anything else. They looked at cytology, Ki67, P53, and SOX11. So how did it look? Well, the Ki67 coefficient was about 0.87 as a continuous parameter. When you dichotomize it at 30%, and you say, is the patient above or below 30%, the agreement's only 65%, which is not too great. It's almost a 50-50 coin flip there. The cases with the discrepant results were usually pretty close to 30%. And I can tell you as a pathologist, when I'm looking at one of these, I'm like, oh, is that 25 or 35? And knowing that it might make a difference in the message that the patient gets makes a big difference, and it is an area of sort of anxiety. They did have it all centralized scored and computer aided, and that seemed to help. So there may be a way to standardize this in the future. But this is just a picture of what the Ki67 staining looks like. One lab stained it and came out kind of light, and another lab stained it and came out really dark. With Ki67 staining, you don't have to look at the intensity. You just have to say if it's positive or negative. So something that's very light, like this, you might be calling positive, or you would want to call positive, but it's a lot easier to call it positive when the stain is a little bit more robust. So that was a figure from the paper. This is the 20 cases with 8 pathologists each. Again, variability here from 35 up to 75, and this is the same field because it's a tissue microarray. Doesn't matter if they're both on the same side of 30. Another case here down at 0 up to 25. They're both on the same side of 30, but these are the problem cases where the counts are ranging from 15 up to 55, or, and they're crossing. So this one patient's case, depending on who looked at it, they may have a different result. Other results, cytology, I was surprised at this. Of the 4 types, the agreement was 39, was 0.39. So we have to do a better job at defining how we look at cytology. P53 is an important marker. It's a 4-tier system. You 0 to 25, 25 to 50, 50 to 50. So that had pretty good agreement. And then the SOX11 positive versus negative, we know that those leukemic non-nodal cases tend not to have SOX11, but that's just is it there, yes or no? It's an all or nothing. And that correlated super well. Pathologists did really well with that 95% agreement. And so, and that correlated with this other expression signature, which I decided not to talk about today, the leukemic non-nodal mantle cell expression signature. So what we found in some other work was that you can take those mantle cells that are circulating in the peripheral blood, and you can define expression signature related to them. If you do that on cases that are in the lymph node, you can sometimes find a leukemic non-nodal type in a lymph node. So that might be something that people would want to know about. So anyway, the final comments were our analysis supports the concept of molecular signatures as valid substitutes, but also we should probably continue to work on standardization and reproducibility. Although for me, somebody who deals with multi-parameter, I always think that just one stain is maybe not going to capture what, looking at 35 different parameters and averaging that all out will do, but what remains to be seen. So what are our next steps? All of the work that I showed you was done on younger patients with R-CHOP or intensified, more intense regimens, dose-intense regimens that were younger. And what we haven't looked at are the older patients, and there's a lot of older patients, older than 65, and right now a lot of them are being treated with Rituxan and bendamustine, so BR or B, whatever you call it. So we wanted to look at a population series, not patients on clinical trials necessarily. They're carefully selected to be very healthy and et cetera. So we want to look at a clinical trial and also at a population series. So I contacted, and I wanted to bring this up, the LEO consortium, and Emory University is part of that. Several of you work in that. Dr. J, we were talking about that, Dr. Mann, and others. And this is a collection of lymphoma cases that get collected, described, the patients are consented, their tissue goes into a tissue bank that is then used, for various projects. And Dr. Cohen, who unfortunately couldn't be here today, was very active in this at this site. And so I went to the Leo, and you were able to provide some cases. I'm going to show the data on that next. And then following that, we'll go to a clinical trial cohort. So this is the first, never before seen, mantle cell of MCL35 in R bendamustine treated patients. Again, Ki-67 versus mantle versus MCL35. Only a few really high-risk patients in this group, and small numbers, only 31. But you can see the Ki-67 spread as well in the intermediate risk as well as the low risk. And some of those, these are low-risk patients by MCL35. They would look high risk by Ki-67. Same thing with these guys. So that would be, I would consider those discrepancies. And then you can see the real scatter here between MCL35 and Ki-67. Not a very good correlation. Again, very low numbers. But then the question was, well, which one is really the better one? And we've done this with the younger patients, but we wanted to look at an older, again, very small numbers. This is the Ki-67, looking at overall survival and event-free survival, and seeing really it's not distinguishing them, and we weren't that surprised to see these data. Looking at the MCL35, though, we were able to see, and again, very few numbers, but we could pick out the high-risk patients that were doing worse versus the others. So we're hoping that by adding to this, we'll be able to really round out the story and say maybe this will be useful in the older patients as well. This data was just submitted to the Lymphoma Research Foundation, the Mantle Cell Lymphoma Workshop, which is going to be in Chicago. In the meantime, we're collecting additional cases. Like I said, Emory University, you guys, Leo supplied 16 of these initial cases, and then we're going to our friends in British Columbia, Nebraska, City of Hope, the NIH, and a few European sites, and collecting additional cases. The U.S. clinical trial we're going to work with, Dr. Brad Call was one of the speakers here, I think 2 years ago. He's one of the clinicians. I talked to him about using MCL35 in the E1411 trial, which is closed. That was a 4-arm phase 2 trial with older patients with previously untreated mantle cell lymphoma. They plan to accrue about 332. They're actually closed to accrual now and waiting on the follow-up data. So we will be looking at this again. Does this help with patients who are ineligible for transplant? And he wrote a letter of support, and he's actually helping me to navigate my way through the National Cancer Institute to get permission to use the samples. Okay, so what other science is going on? Because I didn't want to leave you thinking we spent all our time on proliferation and technology, although it has been a big part of our story. The LLMPP group, which I have mentioned, doing the proliferation signature and then the MCL35, we currently have a PO1 grant, and Project 2 is completely focused on the biology of mantle cell lymphoma, looking at pathogenesis of what we're calling now conventional or classic mantle cell versus leukemic non-nodal and versus blastoid mantle cell lymphoma. And this was kind of an early concept of a couple of different cell types that have translocations of 11-14. You go to an in situ mantle cell lymphoma. Some of them go into the germinal center, become hypermutated, lose SOX11, and then become the leukemic type and progress on to a more aggressive mantle cell lymphoma, versus the other ones that start at in situ, stay in that mantle zone, minimally mutated, are SOX11 positive, and that gives you what we call classical mantle cell lymphoma that can then become blastoid. These are our specific aims. We're 18 months into a 5-year project. And my whole group is meeting at Mayo Clinic in 2 weeks. So we are looking at cyclin D1 and the generation of mantle cell diversity, beyond just cell cycle regulation, we're looking at cyclin D1 and saying, what else does this correlate within the cell? I mean, we know that it has to do with cell cycling and proliferation, but what else, what other processes are being disturbed, and how does it interact with SOX11? And we have some very preliminary data coming out on that is not even in a manuscript stage, just being kicked around. But it looks like there could be some relationships to chromosomal instability and that whole process of maintaining chromosome integrity. We're also going to be looking at other genomic changes, other epigenetic alterations. So epigenetics, looking at the methylation patterns, which genes are turned on and off, and how that might drive clinical biological behavior of conventional versus the non-nodal. And then the AIM-3 will be looking at biomarkers to look at that, the nodal, non-nodal, and improve on that, and maybe also helping us to look for new targetable pathways for those patients who maybe can't get the intense chemotherapy. This was a little bit more detail of what I showed a few minutes ago of a pre-B cell with a translocation going to a naive B cell. The ones that stay in the conventional type, someone was talking to me about BCL6 this morning and MTA3. Anyway, so SOX11 stopping BCL6 and then having effects on the PAX5, FAX, CXCR4, and PDGF alpha, all being part of the lymphoma genesis of conventional mantle cell, and then again the naive B cell going to the germinal center, exiting as a highly mutated cell, becoming that leukemic version, and then both of these, either one gets a TP53, you know, the cell cycle checkpoint, either one of those gets inactivation of that gene, and then you get the blastoid, because they can both turn out to have a very aggressive kind of final type. So this is my translational research program pyramid. And I start at the bottom with the tissue, and this is every single patient who gives their consent and says, my tissue can go into the bank. You can pull it out, we can look at it, we can look at what happened and study this over and over again with more and more sophisticated methods. And so it starts at the operating room and in the patient consultation where the permission is given to harvest that tissue. Then we go to the basic science, immunology, and technology, and you can see I've tried to weave that through the talk, that as things have changed over the years, more and more we've learned. Translational research, this is kind of where I'm living, diagnostic prognostics, target identification, that preclinical. I've also been very lucky to work with SWOG and be part of the clinical trials designs, where all your medical oncologists are taking care of the patients who are at the top. And does anyone remember that horrible old cigarette commercial that said, "We've come a long way, baby"? Do you remember that? And the lady was smoking. So I end up with, this was 50 years and 50 minutes. And we've come a long way since diffuse small cell cell lymphoma in what we've learned, how we define it, and what we know about the biology. And those are my acknowledgments, the people in my lab, the LLMPP, my clinical collaborator, and the Mayo Clinic for the support. So that's the end of today's talk. Thank you.